We're now back with the subject of molecular interactions relevant to cellular biology. And we're on the topic of vitamins, which I last stopped in the previous lecture. So continuing from there, vitamins, number 19. Both vitamins and minerals are needed by the body in very small amounts to trigger the thousands of chemical reactions necessary to maintain good health. Many of these reactions are linked with one triggering another. If there is a missing or deficient vitamin or mineral or link anywhere in this chain, this process may break down with potentially devastating health effects. Although similar in supporting critical functions in the human body, vitamins and minerals have key differences. Among their many functions, vitamins enhance the body's use of carbohydrates, proteins, and fats. They are critical in the formation of blood cells, hormones, nervous system chemicals known as neurotransmitters, and the genetic material deoxyribonucleic acid, DNA. Vitamins are classified into two groups, fat-soluble and water-soluble. Fat-soluble vitamins, which include vitamins A, D, E, and K, are usually absorbed with the help of food that contain fat. Fat containing these vitamins is broken down by bile, a liquid released by the liver, and the body then absorbs the breakdown products. And unsaturated fats has also been associated with greater risk of developing cancers of the colon, prostate, breast, and uterus. Choosing a diet that is low in fat and cholesterol is critical to maintaining health and reducing the risk of life-threatening diseases. Excess amounts of fat-soluble vitamins are stored in the body's fat, liver, and kidneys. Because of these vitamins can be stored in the body, they do not need to be consumed every day to meet the body's needs. Water-soluble vitamins, which include vitamin C, also known as ascorbic acid, B1 thiamine, B2 riboflavin, B3 niacin, B6 B12, and folic acid, cannot be stored and rapidly leave the body in urine if taken in greater quantities than the body can use. Foods that can contain water-soluble vitamins need to be eaten daily to replenish the body's needs. In addition to the roles noted in the vitamin and mineral charts accompanying this article, vitamins A in the form of beta-carotene, C and E function as antioxidants, which are vital in countering the potential harm as chemicals known as free radicals. If these chemicals remain unchecked, they can make cells more vulnerable to cancer-causing substances. Free radicals can also transform chemicals in the body into cancer-causing. That's just a table there. We'll come back. And now onto a table of all the vitamins and details therein. Vitamin A is called retinol. It's a yellow viscous oil. Its function would be anti-xerophthalmic, synthesis of retinal pigments, maintenance of epithelia, prevention of keratinization, anti-infective. Deficiency would lead to xerophthalmia, night blindness, keratomalacia, dermatosis, toad skin, and stunted growth. Source would be carotene in vegetables, mango, orange, and as such in milk, butter, ghee, egg yolk, and liver oil. Next would be D-sterol, vitamin D, sterol calciferol, 10 types, common D2 and D3, anti-rachitic, absorption and retention of calcium and phosphorus, rickets in children, osteomalacia in adults, and sunshine is the source. Next will be E, phocoferol, colorless oil alpha and beta, anti-sterility factor, anti-oxidative for membranes, lipids, reduces keratinization, and hair reduces atherosclerosis. It causes its lack or deficiency causes erythrocyte breakdown, anemia, muscular dystrophy, cramps, miscarriage, and reduced fertility. Sources would be green vegetables, oils, milk, cheese, butter, and eggs. The next would be vitamin K, also called as phylloquinone. It's a yellow fat and oil, K1 and K2. Anti-hemorrhagic factor for synthesis of prothrombin, hence essential for blood coagulation. Electron carrier, liver functioning. It causes deficiency causes prolonged bleeding, disease, or hemorrhages, liver malfunctioning. Its sources would be green vegetables, tomato, and colon bacteria. The next would be vitamin thiamine B1 and vitamin G. anti-neurotic, anti-beriberi, component of TPP, functioning of muscles and nerve are its functions. Its deficiency causes anorexia, muscular atrophy, beriberi in humans, and polyneuritis in animals. Its sources would be yeast, whole eggs, egg yolk, and liver. And the next would be riboflavin, B2, maintenance of skin and mucosa, component of FMN and FAD anti-migraine. Key losses, glossitis and intolerance to light would be the outcome of its deficiency, and its sources would be yeast, green vegetables, milk, curd, egg, and cheese. Next would be pantothenic acid, which would be known as B3, also B5, formation of CoA, required in cell respiration, functioning of skin and nerves, gastrointestinal disorders, graying of hair in animals, burning feet syndrome. Its sources would be yeast, milk, wheat bran, pulses, liver, colon bacteria. The next would be nicotinamide of niacin and nicotinic acid, N6, also B5, B7, vitamin POP, pellagra preventive. Anti-pellagra, a component of NAD+, NADP+, energy transfer and nervous system. Its deficiency causes pellagra. Its source is yeast, pulses, green vegetables, cereals, milk, egg, yolk, and liver. Pyridoxine, B6, component of coenzyme pyridoxal phosphate, are its functions. And its deficiency, it would cause anemia, neuritic pain, convulsions, skin lesions. Its sources would be yeast, green vegetables, egg yolk, wheat, germ, and liver. Next will be biotin, B4, which is also known as vitamin H. Prostatic group of some enzymes in energy liberation and fat synthesis. Avid and raggig, white prevents biotin absorption. Its deficiency causes dermatitis, anorexia, vascular pain, rise in blood cholesterol. Its sources would be yeast, vegetable fruits, egg yolk, colon, and bacteria. The next would be folic acid, vitamin M, and pteroglutamic acid. It's required in erythrocyte formation, in DNA and RNA synthesis, and helps in transfer of methyl groups. Its deficiency causes megaloblastic anemia, gastrointestinal disorders, and glossitis. And its sources would be leafy vegetables, egg yolk, and colon bacteria. The next will be cobalamin, B12, which is required in erythrocyte maturation, DNA and RNA synthesis, pernicious anemia, glossitis, loss of peripheral sensation. Sources would be milk, egg, liver, fish, and colon bacteria. And next will be vitamin C or ascorbic acid, heat bile. Absorption of iron, formation of dentine, collagen, and bone matrix amino acid. And oxidation of matric bone amino acids. And next will be its deficiency that leads to scurvy, stiff sore joints, fragile bone, leaves, bone vessels and bones, delayed wound healing. And it is sourced from citrus fruits, guava, tomato, amla, that's gooseberry, and mangoes. The next portion will be on non-essential nutrients, which stands at number 20, that is cholesterol. Cholesterol, like fat, is a lipid and organic compound that is not soluble in water. In order to travel through blood, cholesterol therefore must be transported through the body in special carriers called lipoproteins. High-density lipoproteins, HDLs, remove cholesterol from the walls of arteries, return it to the liver, and help the liver excrete it as bile, a liquid acid essential to fat digestion. For this reason, HDL is called good cholesterol. Low-density lipoproteins, LDLs, and very low-density lipoproteins, VLDLs, are considered bad cholesterol. Both LDLs and VLDLs transport cholesterol from the liver to the cells. As they work, LDLs and VLDLs leave plaque-forming cholesterol in the walls of the arteries, clogging the artery walls and setting the stage for heart disease. Almost 70% of the cholesterol in our bodies is carried by LDLs and VLDLs. and the remainder is transported by HDLs. For this reason, we need to consume dietary fats that increase our HDLs and decrease our LDL and VLDL levels. The next will be 21. That's about saturated fatty acids. All right. Found in foods ranging from beef to ice cream to mozzarella, cheese to donuts should make up no more than 10% of a person's total calorie intake each day. Saturated fats are considered harmful to the heart and blood vessels because they are thought to increase the levels of LDLs and VLDLs and decrease the levels of HDLs. Monounsaturated fats found in olive, canola, and peanut oils appear to have the best effect on blood cholesterol, decreasing the level of LDLs and VLDLs and increase the level of HDLs. Polyunsaturated fats found in margarine and sunflower, soybean, corn, and safflower oils are considered more healthful than saturated fats. However, if consumed in excess, more than 10% of daily calories, they can decrease the blood levels of HDLs. Next will be nutritional disorders. 21. That's marasmus. It is a disorder caused by deficiency of food or protein energy malnutrition, PEM. Marasmus occurs in infants 1 to 2 years when they do not get sufficient mother's milk and weaning diet. Such infants are often provided with much diluted cow or buffalo milk. Marasmus babies have shriveled appearance with thin face, sunken eyes, wrinkled and dry skin, protruding ribs, reduced digestion, and frequent diarrhea, low weight, little growth, and poor mental abilities. 22 will be kwashiorkor. It is a protein energy malnutrition, PEM, that occurs in children of 1 to 4 years due to constant reduced protein in diet. This can be due to 1. Early stoppage of breast milk, 2. Late supply of supplementary food. A third would be over-diluted milk. A fourth would be maintaining children on cooking water of cereals. A kwashiorkor child has matchstick legs, protruded belly, a blotchy skin, anemia, edema of certain parts, and repeated diarrhea. The next will be obesity. Obesity is a luxury nutritional disorder caused by a greater intake of food than the requirement of the body. It is quite common in persons having higher intake of sweets, carbohydrates-rich food, fried articles, fats-rich food, and absence of roughage in food. Obese persons are overweight with excessive accumulation of fat. Moments are reduced. There is a risk of diabetes, osteoarthritis, gallbladder stones, high blood pressure, and cardiac problems. The reasons for overeating are psychogenic, physiological, genetic, and developmental. The next will be hypercholesterolemia. It is characterized by high blood cholesterol due to a high intake of cholesterol-rich eggs and meat, butter, and ghee, or reduced iodine intake. Cholesterol gets deposited on the walls of blood vessels, making their lumen narrow and walls stiffer. This increases blood pressure or hypertension. It leads to cardiovascular disorders. And the 25th would be rickets. The disorder is due to deficiency or deficient ossification of bone that leads to the bending and swelling of joints, where the knee, wrist, and elbow. It occurs in children of 6 months to 2 years when the diet is deficient in vitamin D, calcium, or phosphorus. Osteomalacia, 26. Osteomalacia, the disorder occurs in adults when their food is deficient in vitamin D, calcium, or phosphorus. Bones of vertebral column become weak. Pelvic and other bones bend and become soft. So xerophthalmia is the next. It is a vitamin A deficiency disorder which is caused by stoppage of lacrimal activity. Cornea and eyelids become dry and ulcerated. It leads to swelling of dry eyelids and opacity of cornea. Xerophthalmia, if untreated, leads to blindness. Next topic will be night blindness, nyctalopia. Dietary deficiency of vitamin A results in deficiency of rhodopsin or visual purple. The latter impairs the ability to see in dim light at night. Beriberi is a debility produced by deficiency of thiamine. As during excessive intake of polished rice, it results in loss of appetite, anorexia, weakening of muscles, fatigue, poor digestion, palpitations, slow reflexes, body rigidity and neuritis. There is inflammation of peripheral nerves, weakening of muscles, paralysis, and progressive edema. Depending upon the system affected, beriberi is of 5 types: dry, wet, mixed, cardiac, and cerebral. The next will be scurvy or sailor's disease. Vitamin C ascorbic acid deficiency leads to scurvy, characterized by bleeding gums, falling of teeth, fragile blood vessels, fragile bones, and slow healing of injuries. The next would be anemia, which is a disorder characterized by reduction in hemoglobin content of the blood due to either few red blood corpuscles or their hemoglobin content. It is of 3 types: microcytic, most common due to iron deficiency leading to fewer and smaller erythrocytes with reduced hemoglobin; megablastic due to fewer abnormal red blood cells caused by deficiency of folic acid or B12; pernicious due to production of hemoglobin-free mature RBCs caused by B12 deficiency as a result of deficient intrinsic factor; sickle cell anemia, thalassemia genetically controlled. Anemia also occurs due to excessive blood loss. The same is called post-hemorrhagic anemia. We move to the next topic on minerals. Minerals are sodium, chlorine, potassium, magnesium, sulfur, cobalt. Right? And sodium, it is the main cation of extracellular fluid. Involved in HCl synthesis and acid-base balances. Let's go back again. Sodium, it is the main cation of extracellular fluid, component of bile salts, which is involved in osmotic balance, acid-base balance, absorption of glucose, electrochemical impulse conduction in nerves and muscles. Deficiency produces cramps. Next is chlorine, it main anion of extracellular fluid, involved in HCl synthesis and acid-base balance. Potassium, the cation is present in intracellular as well as extracellular fluid. It takes part in muscle and nerve activity, glycogen protein synthesis. Magnesium, it is an enzyme activator, component of bones and teeth besides being required for muscle relaxation. Deficiency produces convulsions. Sulfur, it is a constituent of many proteins, enzymes, and coenzymes. And cobalt, a component of B12, deficiency causes pernicious anemia. And fluorine. Fluorine, it maintains enamel and checks dental decay. It's harmful to teeth and bones. The calcium, it is a major component of bones and teeth. Calcium is also required for blood clotting, muscle contraction, ATPase, nerve impulse transmission, heart functioning, etc. Vitamin D is essential for calcium absorption. Deficiency produces rickets and muscle spasms. Phosphorus, along with calcium, it occurs in bones and teeth. Phosphorus is a component of nucleic acids. Phospholipids A, T, P, and some coenzymes. Phospholipids, ATP, and some coenzymes. Phosphorylation activates sugars. And fatty acids. Phosphate is pH buffer. Deficiency reduces growth, metabolism, and causes rickets in children, along with calcium and vitamin D. Iron, it is reused in the body. Iron is a component of hemoglobin, myoglobin, cytochromes, and pyridoxine. Deficiency leads to anemia. Iodine, it is essential for production of a hormone thyroxine of thyroid glands. Deficiency causes goiter. Selenium, essential for protecting sperms and hence maintenance of male fertility. Stabilizing reactions. The covalent bonds are very stable because the energies required to break them are much greater than the thermal energy available at room temperature, 25 degrees centigrade, or body temperature, 37 degrees centigrade. For example, the thermal energy at 25 degrees centigrade is approximately 0.6 calorie per mole or kilocalories per mole, whereas the energy required to break the carbon-carbon single bond, CC, in ethane is about 140 times larger. Consequently, at room temperature, 25 degrees centigrade, fewer than 1 in 10 raised to 12 ethane molecules is broken into a pair of CH3 radicals, each containing an unpaired non-bonding electron. Covalent single bonds in biological molecules have energies similar to that of the CC bond in ethane. Because more electrons are shared between atoms in double bonds, they require more energy to break than And single bonds, for instance, it takes 84 kilocals per mole to break a single CC bond, but 170 kilocals per mole to break a CO and double bond. The most common double bonds in biological molecules are C double bond O, C double bond N, C double bond T, and P double bond O. See the notations. The energy required to break non-covalent interactions is only 1 to 5 kilocalories per mole, much less than the bond energies of covalent bonds. Indeed, non-covalent interactions are weak enough that they are constantly being formed and broken at room temperature. Although these interactions are weak and have a transient existence at physiological temperatures of 25 to 37 degrees centigrade, multiple non-covalent interactions can act together to produce highly stable and specific associations between different parts of a large molecule or between different macromolecules. We first review the 4 main types of non-covalent interactions and then consider their role in the binding of biomolecules to one another and to other molecules. This is a diagram of that, non-covalent interactions, where the electrostatic, van der Waals, thermal energy, hydrogen bonds, hydrolysis of phosphine hydrate takes place, and van der Waals interactions. When any two atoms approach each other closely, they create a weak, non-specific attractive force that produces a van der Waals attraction, named for Dutch physicist Johannes Dietrich van der Waals, 1837 and 1923, who first described it. These non-specific interactions result from the momentary random fluctuations in the distribution of electrons of any atom, which give rise to a transient unequal distribution of electrons, that is, a transient electric dipole. If two non-covalently bonded atoms are close enough together, the transient dipole in one atom will perturb the electron cloud of the other. This perturbation generates a transient dipole of the second atom, and the two dipoles will attract each other. Similarly, a polar covalent bond in one molecule will attract an oppositely oriented dipole in another. Van der Waals interactions. Van der Waals interactions. When any two atoms approach each other closely, they create a weak, non-specific attractive force that produces a van der Waals interaction, named for Dutch physicist Johannes Dietrich van der Waals, 1837 to 1923. Who first described it. These non-specific interactions result from the momentary random fluctuations in the distribution of the electrons of any atom, which give rise to a transient unequal distribution of electrons, that is, a transient electric dipole. If non-covalently bonded atoms are close enough together, the transient dipole in one atom will perturb the electron cloud of the other. This perturbation generally generates a transient dipole in the second atom, and two dipoles will attract each other weakly. Similarly, a polar covalent bond in one molecule will attract an oppositely oriented dipole in another. And these non-specific interactions result from the momentary random fluctuations in the distribution of electrons of any atom, and which gives rise to a transient electric dipole. If two non-covalently bonded atoms are close enough together, the transient dipole in one atom would perturb the electron cloud of the other. This perturbation generates a transient dipole in the second atom, and the two dipoles will attract each other weakly. Similarly, a polar covalent bond in one molecule will attract an oppositely oriented dipole in another. Van der Waals interactions involving either transient-induced or permanent electric dipoles occur in all types of molecules, both polar and nonpolar. In particular, van der Waals interactions are responsible for the cohesion between molecules of nonpolar liquids and solids, such as heptane, CH3, CH2, CH3, that cannot form hydrogen bonds or ionic interaction with other molecules. When these strong interactions are present, they override most of the influence of van der Waals interactions. Heptase, however, would be a gas if van der Waals interactions could not form. The strength of van der Waals interactions decreases rapidly with increasing distance. Thus, these non-covalent bonds can form only when atoms are quite close to one another. However, if atoms get too close together, they become repelled by the negative charges in their outer electron shells. When the van der Waals attraction between two atoms exactly balances the repulsion between the two electron clouds, and the atoms are said to be in van der Waals contact. The diagram here depicts that of repulsion energy with attraction. So van der Waals contact distance, and here's the distance covered. So energy of van der Waals interactions as a function of the distance between atoms. So each type of atom has a van der Waals radius at which it is in van der Waals contact with other atoms. The van der Waals radius of an H atom is 0.1 nanometer, and the radii of oxygen, nitrogen, carbon, and sulfur atoms are between 0.14 and 0.18 nanometer. Two covalently bonded atoms are closer together than two atoms that are merely in van der Waals contact. For a van der Waals interaction, the intercellular distance is approximately the sum of the corresponding radii for the two participating atoms. Thus, the distance between a carbon atom and an H atom in van der Waals contact is 0.27 nanometer, and between two C atoms is 0.34 nanometer. In general, the van der Waals radius of an atom is about twice as long as its covalent radius. For example, a CH covalent bond is about 0.107 nanometer long, and a CC covalent bond is about 0.154 nanometer long. Note these diagrams here. The covalent radius is 0.062 nanometers, whereas the van der Waals radius is 0.14. See, the distance of interaction, right? The energy of the van der Waals interaction is about 1 kilocalories per mole, only slightly higher than the average thermal energy of molecules at 25 degrees centigrade. Van der Waals interactions, as well as other non-covalent bonds, mediate the binding of many enzymes with a specific substrate, the substances on which an enzyme acts, and of each type of antibody with its specific antigens. C2. Electrostatic ionic interactions. In some compounds, the bonded atoms are so different in electronegativity that the bonding electrons are never shared. These electrons are always found around the more electronegative atom. In sodium chloride, sodium NaCl, for example, the bonding electron contributed by the sodium atom is completely transferred to the chlorine atom. Even in solid crystal of sodium chloride NaCl, the sodium and chlorine atoms are ionized, so it is more accurate to write the formula for the compound as sodium and chlorine. Because the electrons are not shared, the bonds in such compounds cannot be considered covalent. They are rather ionic bonds or interactions that result from attraction of a positively charged ion, a cation, for a negatively charged ion and an anion. Unlike covalent or hydrogen bonds, ionic bonds do not have fixed or specific geometric orientations because the electrostatic field around an ion, its attraction for an opposite charge, is uniform in all directions. However, crystals of salts such as sodium and chlorine do have very regular structures because that is the energetically most favorable way of packing together positive and negative ions. The force that stabilizes ionic crystals is called the lattice energy. In aqueous solutions, simple ions of biological significance such as sodium, potassium, calcium, magnesium, and chlorine do not exist as free, isolated entities. Instead, each is surrounded by a stable, tightly held shell of water molecules. An ion interaction occurs between the ion and the oppositely charged end of the water dipole, as shown below for the K ion. Ions play an important biological role when they pass through narrow protein-lined pores or channels in membranes. For example, ionic movements through membranes are essential for the conduction of nerve impulses and for the simulation of muscle contraction. The ions must lose their shell of water molecules in order to pass through the ion channel proteins. Channel proteins can then selectively admit only sodium or potassium or calcium ions, a selectivity essential for nerve function. Most ionic compounds are soluble in water because a large amount of energy is released when the ions tightly bind water molecules. This is known as the energy of hydration. Oppositely charged ions are shielded from one another by the water and tend not to recombine. Salts like sodium, chlorine dissolve in water because the energy of hydration is greater than the latter's energy that stabilizes the crystal structure. All right, so that's a good reasoning there about how it dissolves. Salts like sodium and chlorine dissolve in water because the energy of hydration is greater than the latter's energy that stabilizes the crystal structure. In contrast, certain salts such as calcium and phosphate, are virtually insoluble in water. The larger charges on the cation, on the calcium ion and phosphate ions, generate a formidable lattice energy that is greater than the energy of hydration. That's the difference. Some are soluble and not soluble, right? So C3, the hydrogen bond. Normally, a hydrogen atom forms a covalent bond with only one other atom. However, a hydrogen atom covalently bonded to a donor atom D may form an additionally weak association with the hydrogen bond with an acceptor atom D8, that's deuterium, to hydrogen delta plus A delta, reversibly gives D delta to H delta as a hydrogen bond with A delta. In order for a hydrogen bond to form, donor atom must be electronegative so that the covalent DH bond is polar. The acceptor atom also must be electronegative, and its outer shell must have at least one non-bonding pair of electrons that attracts the D plus charge of the hydrogen atom. In biological systems, both donors and acceptors are usually nitrogen or oxygen atoms, especially those atoms in amino, NH2 and hydroxyl OH groups. Because all covalent NH and OH bonds are polar, the H atoms can participate in hydrogen bonds. By contrast, CH bonds are nonpolar, so these H atoms are almost never involved in a hydrogen bond. Water molecules in biological systems, both donors and acceptors are usually nitrogen or oxygen atoms, especially those atoms in amino hydroxyl groups. Because of all covalent NH and OH bonds are polar, the H atoms can participate in hydrogen bonds. By contrast, CH bonds are nonpolar, so these H atoms are almost never involved in a hydrogen bond. Water molecules provide a classic example of hydrogen bonding. The hydrogen in one water molecule is attracted to a pair of electrons in the outer shell of an oxygen atom in an adjacent molecule. So this is how it is. A water molecule is being connected, right? In general, molecules with water can dissolve in water and are said to be hydrophilic, Greek, water-loving. Besides the hydroxyl and amino groups, peptide and ester bonds are important chemical groups that interact well with water. Most hydrogen bonds are 0.26 to 0.31 nanometer long, about twice the length of a covalent bond between these same atoms. In particular, the distance between the nuclei of the hydrogen and oxygen atoms of adjacent hydrogen-bonded molecules in water is approximately 0.27 nanometer, about twice the length of the covalent OH bonds in water. The hydrogen atom is closer to the donor atom D, to which it remains covalently bonded, than it is to the acceptor. The length of the covalent DH bond is a bit longer than it would be if there were no hydrogen bonds, because the acceptor pulls the hydrogen away from the donor. The strength of a hydrogen bond in water is double squabbled 5. Approximately 5 kilocals per mole is much weaker than a covalent OH bond, approximately 110 kilocals per mole. Hydrogen bond is a major stabilizing force in macromolecules. Is an important answer there. We're going on to the next page. An important feature of all hydrogen bonds is directionality. In the strongest hydrogen bonds, the donor atoms, the hydrogen atom, and the acceptor atom all lie in a straight line. Nonlinear hydrogen bonds are weaker than linear ones. The strengths of the hydrogen bonds in proteins and nucleic acids are only 1 to 2 kilocals per mole, considerably weaker than the hydrogen bonds between water molecules. Initially, both the OH and NH2 groups in the protein are hydrogen bonded to water, and the formation of a hydrogen bond between these groups involves disruption of their hydrogen bonds with water. Thus, the net change in energy in forming this OH-N hydrogen bond will be less than the 5 kilocalories per mole characteristic of hydrogen bonds between water molecules. The next topic will be hydrophobic interaction. Nonpolar molecules do not contain ions because a dipole movement or become hydrated because such molecules are insoluble or almost insoluble in water. They are said to be hydrophobic. Greek is water-fearing. The equivalent bonds between two carbon atoms and between carbon and hydrogen atoms are the most nonpolar bonds in biological systems. Hydrocarbons, or molecules made up only of carbon and hydrogen, are virtually insoluble in water. A large triacylglycerol or triglycerides such as tristyrene, a component of animal fat, is also insoluble in water, even though its 6 oxygen atoms participate in some slightly polar bonds with adjacent carbon atoms. When shaken in water, tristyrene forms a separate phase similar to the separation of oil from the water-based vinegar in an oil and vinegar salad dressing. Note this diagram. You have the unaggregated state, water population highly ordered, right? Lower entropy, energetically unfavorable. But you see this diagram, the water releases into bulk solution, where aggregated state, water population less ordered, higher entropy, and energetically more favorable. This and this. Make a screenshot of this. You need it as a reference, right? And then going down here. The forces that causes hydrophobic molecules or nonpolar portions of molecules to aggregate together rather than to dissolve in water is called the hydrophobic bond. This is not a separate bonding force, rather it is a result of the energy required to insert a nonpolar molecule into water. A nonpolar molecule cannot form hydrogen bonds with water molecules, so it distorts the usual water structure, forcing the water into a rigid cage of hydrogen-bonded molecules around it. Water molecules are normally in constant motion, and the formation of such cages restricts the motion of a number of water molecules. The effect is to increase the structural organization of water. This situation is energetically unfavorable because it decreases the randomness entropy of the population of water molecules. The opposition of water molecules to having their motion restricted by forming cages around hydrophobic molecules, or portions thereof, is the major reason molecules such as tristyrene and heptane are essentially insoluble in water and interact mainly with the other hydrophobic molecules. Nonpolar molecules can also bond together, albeit weakly, through van der Waals interactions. The net result of the hydrophobic and van der Waals reaction is very powerful tendency for hydrophobic molecules to interact with one another and not with water. The order of hydrophobic character for various amino acids is as follows. Hydrophobic, glycine to tyrine, alanine. Let's repeat that again. Glycine less than tyrine, less than alanine, less than histidine, less than methane, less than valine, less than leucine, less than isoleucine. I think it's leucine. Less than phenylalanine and tryptophan, less than tryptophan. Small hydrocarbons like butane, CH3, CH2, CH2, CH2 single bonds are somewhat soluble in water because they can dissolve without disrupting the water lattice appreciably. However, 1-butanol CH3, CH2, CH2, CH2OH mixes completely with water in all proportions. The replacement of just one hydrogen atom with a polar OH group allows the molecule to form hydrogen bonds with water and greatly increases its solubility. Simply put, dissolves like polar molecules dissolve in polar solvents such as water, while nonpolar molecules dissolve in nonpolar solvents such as hexane. Now, molecular complementary permits tight, highly specific binding of biomolecules. Both inside and outside of cells, ions and molecules are constantly bumping into one another. The greater the number of copies of any two types of molecules per unit volume, that is, the higher their concentration, the more likely they are to encounter one another. When two molecules encounter each other, they most likely will simply bounce apart because the non-covalent interaction that would bind them together are weak and have a transient existence at physiological temperatures. right? However, molecules that exhibit molecular complementarity, a lock and key kind of fit between the shapes, charges, or other physical properties can form multiple non-covalent interactions at close range. When two such structurally complementary molecules bump into each other, they can bind or stick together. Note this diagram. Just magnify it for you. Have a look at this. Proteins, the negative and positives are attracted here. The ionic bonds, the hydrogen bonds, hydrophobic and van der Waals interactions, clearly shown here. Protein A, protein B stable complex, protein A, protein C less stable complex. You see that? So the figure illustrates how multiple different weak bonds can bind two proteins together. Right? Almost any other arrangement of the same group on the two surfaces would allow the molecules to bind so tightly. Such simple specific interactions between complementary regions within a protein molecule allow it to fold into a unique three-dimensional shape and hold the two chains of DNA together in a double helix. Similar interactions underlie the association of groups of more than two molecules into multimolecular complexes, leading to formation of muscle fibers, to the glue-like associations between cells and solid tissues, and to numerous other cellular structures. Depending on the number and strength of the non-covalent interactions between the two molecules and on the environment, the binding may be tight, strong, or loose, weak, and as a consequence, either long-lasting or transient. The higher the affinity of two molecules for each other, the better the molecular fit between them, and the more non-covalent interactions can form, and the tighter they can bind together. An important quantitative measure of affinity is the binding dissociation constant, KD. Nearly all chemical reactions that occur in cells can also depend on the binding properties of enzymes. These proteins not only speed up reactions, but also do so with a higher degree of specificity, a reflection of the ability to bind tightly to not only one or a few related molecules. Indeed, the binding specificity of large molecules, biological molecules, particularly proteins and nucleic acids, is one of the distinct features that distinguish biochemistry from typical solution chemistry. Clearly, molecular complementarity and non-covalent interactions underlie the structures of biomolecules and many processes critical to life. Principles of biophysical chemistry: water, pH, and buffers. Water is essential to biochemistry because biological macromolecules assume specific shapes in response to the chemical and physical properties of water. Biological molecules undergo chemical reactions in an aqueous environment. Water is a key reactant in many reactions, usually in the form of H+ and OH+ ions. And going further, water is oxidized in photosynthesis to produce molecular oxygen. Oxygen is part of the process that converts the sun's energy into usable chemical form. Expenditure of that energy under aerobic conditions leads to the reduction of oxygen back to water. Physical properties of water. The structure of water closely approximates a tetrahedron with its two hydrogen atoms and the two lone pairs of electrons of its oxygen atom occupying the vertices of the tetrahedron. The high electronegativity of oxygen relative to hydrogen results in the establishment of a permanent dipole in water molecules. The polar nature of water results in negative portions of the molecule being attracted to the positive portions of neighboring water molecules by a largely electrostatic interaction known as the hydrogen bond. Hydrogen bonds are represented as DHA, that's the bonding, where DH is a weakly acidic compound, so the hydrogen atom has a partial positive charge, and A is a weakly basic group that bears lone pairs of electrons. A is often an oxygen atom or a nitrogen atom, occasionally sulfur. 5. Water is strongly hydrogen bonded with each other, each water molecule participating in 4 hydrogen bonds with its neighbors, 2 of which it donates and 2 in which it accepts. Hydrogen bonds commonly form 2 between water molecules and the polar function groups of biomolecules, or between the polar functional groups themselves. The strongly hydrogen bonded character of water is responsible for many of its characteristic properties, most notably as a high heat diffusion, which allows water to act as a heat sink, such that greater heat loss is necessary for freezing of water to other substances. Right? Of other similar molecular mass. A high heat of vaporization, such a relatively more heat must be input to vaporize water compared to other substances of similar molecules or similar molecular mass. An ability to dissolve most polar compounds. An open structure makes ice less dense than liquid water, thereby making ice float, insulating the water beneath it and inhibiting total freezing of larger bodies of water. A variety of weak electrostatic interactions are critical to the structure and reactivity of biological molecules. These interactions include, in order of increasing strength, London dispersion forces, dipole-dipole interactions, hydrogen bonds, and ionic interactions. Water is an excellent solvent of polar and ionic substances due to its property of surrounding polar molecules and ions with oriented shells of water, thereby attenuating the electrostatic forces between these molecules and ions. The tendency of water to minimize its contact with non-polar hydrophobic molecules is called the hydrophobic effect. This effect is largely driven by the increasing entropy caused by the necessity for water to order itself around non-polar molecules. This causes the non-polar molecules to aggregate, thereby reducing the surface area that water must order itself about. Consequently, non-polar substances are poorly soluble in aqueous solution. Many biological molecules have both polar or charged and non-polar functional groups and are therefore simultaneously hydrophilic and hydrophobic. These molecules are said to be amphiphilic, or amphipathic. Dipole moment. You see this, the degree between two hydrogen atoms and an oxygen atom is 104.5 degrees, right? That's a dipole moment. Osmosis is the most important of solvent across a semi-permeable membrane from a region of lower concentration of solute to a region of higher concentration of solute. Osmotic pressure of a solution is the pressure that must be applied to the solution to prevent an inflow of solvent. Hence, an increase in solute concentration results in an increase in osmotic pressure. Diffusion is a random movement of molecules in solution or in the gas phase. It is responsible for the movement of solutes from a region of higher concentration to a region of lower concentration. Chemical properties of water. Water is a neutral polar molecule that has a slight tendency to ionize into H plus and OH minus. However, the proton is never free and binds to a water molecule to form H3O, hydronium ion. The ionization of water is described as an equilibrium between the unionized reactant and its ionized species products. Right? This is a reaction in which H2O irreversibly to H and OH. The constant is equal to H and OH by H2O. Since in dilute aqueous solution, the concentration of water is essentially constant, 55.5 m. The concentration of H2O is incorporated into the value of K, which is referred to as Kw, the ionization constant of water. K14. K of water, not 14. K of water, the constant is equal to H and OH. The values of both H and K are inconveniently small, hence the values are more conveniently expressed as negative logarithms, so that pH is equal to negative log H plus and pK is equal to negative log K. According to Bronsted-Lowry definition, an acid is a substance that can donate a proton and a base is a substance that can accept a proton. The strength of a weak acid is proportional to its dissociation constant, which is expressed as K is equal to HNA by HA. The pH of a solution of a weak acid is determined by the relative concentrations of the acid and its conjugate base. The equilibrium expression for the dissociation of a weak acid can be rearranged to pH is equal to pK plus log A by HA. This relationship is known as Henderson-Hasselbalch equation. When the concentration of a weak acid is equal to the concentration of its conjugate base, pH is equal to pK. Hence, the stronger the acid, the lower its pK. Solutions of a weak acid at pHs near its pK resist large changes in pH as OH or H+ is added. Added protons react with the weak acid's conjugate base to reform the weak acid, whereas base and water added OH combines with the acid to form its conjugate base in water. Solution of a weak acid and its conjugate base in the form of salt is referred to as a buffer. Buffer is effective within 1 pH unit of the pK of the component acid. The properties of most proteins and enzymes, for example, are sensitive to pH as the pH drops. H+ bind to the carboxyl group COO of aspartic acid ASP and glutamic acid GLU, neutralizing the negative effect, and H+ bind to the unoccupied pair of electrons on the N atom of the amino NH2 groups of lysine LYH and arginine ARG, giving them a positive charge. The result: not only does the net charge on the molecule change, it becomes more positive, but many of the opportunities that its R groups have for ionic electrostatic interactions with other molecules and ions are altered. As the pH rises, H+ are removed from the COOH groups of aspartate and glutarate, I guess it's GLU, giving them a negative charge COO, and H+ are removed from the NH3 groups of lysine arginine, removing that positive charge. The result, again, the net charge on the molecule changes. It becomes more negative, and again, many of the opportunities its R groups have for electrostatic interactions with other molecules or ions are altered. The pH of cytosol within a human cell is about 7.4 BAT. This value masks the pH differences that are found in various compartments within the cell. For example, the interior lysosomes is much more acidic, as low as pH 4 to 5 than the cytosol, and the enzymes within work best at these low pH values. The pH differential creator within chloroplasts by the energy of the sun is harnessed to synthesize ATP, which in turn powers the synthesis of food. The pH differential created within mitochondria during the respiration of food is harnessed to the synthesis of ATP, which in turn powers most of the energy-consuming activities of the cell, such as locomotion and biosynthesis of cell components. A tenfold change in H+ alters the pH by 1 unit. Buffers. A buffer mixture is a mixture of a weak acid and the salt of that acid. The practical buffer zone is pKa plus or minus 1 pH. The Henderson-Hasselbalch equation is used for calculations involving buffers. So pH is equal to pKa plus log A minus by HA. pH of a buffer is theoretically independent of dilution, depends only on the ratio of A by HA and pKa. Solving pH problems is the next one. Strong acids completely dissociated. H+ is equal to normality of acid, pH is equal to minus log H+. Strong bases completely dissociated, OH equal to normality of base, POH is equal to minus negative log OH. pH plus POH is equal to 14. Weak acids, two kind of problems. That is, A is the percentage of ionization into molarity is equal to H+. The constant A equal to H, and A by HA equal to X squared by HA square X squared by HA, and X is equal to H+, which is equal to constant A into HA. That's it about that, and we're going to the next topic called Kinetics Dissociation and Association constants. Right? The branch of physical chemistry in which we study rate of reaction mechanism of the reaction is known as chemical kinetics. Rate of reaction. The rate of the reaction may be defined as the rate of change or the concentration of the reactants with time. In other words, it is measured by the amount of substance in unit volume change in unit time, and amount is measured in mole. Thus, rate of reaction is equal to amount transformed by time taken into transformation is equal to dx by dt. That's a differential, where dx is the amount of substance changed and dt is a small interval of time. Negative sign indicates that velocity of the reaction decreases with time. It is important to note that the concept of mechanical velocity or speed cannot be used in measuring the rate of reaction, because rate of reaction depends upon the molar concentrations of the reactants and it decreases with time. The rate of reaction varies with time. The factors influencing the rate of reaction are the effective concentration. The rate of a reaction decreases with the decrease of concentration. The effective temperature. It is observed that rate of reaction increase with rise of temperature. In general, rate of a reaction becomes double on rise of 10 degrees centigrade, usually 25 to 35 degrees centigrade. Effect of the nature of reactant. It is observed that the reactions in which simple ions are involved take place faster than the reactions in which ions of considerable bonds are involved. For example, oxidation Fe2+ by permanganate ion MnO4 ion in acidic medium takes place faster than the oxidation of C2O42- ion under same conditions. Effective catalyst. In general, catalyst increases the rate of reaction. Effective surface area of reactants. It is observed that the smaller particles react more rapidly than the larger particles of the same mass. For example, coal dust bombs rapidly than the large lumps of coal. Coal dust lumps rapidly than the large lumps of coal. All right. Effective radiation. Rate of certain reactions